I am pretty sure I will butcher this last name, so I'm not going to try to say it. But our next speaker is Irena U. Title is Distribution of MAP2K1 variants across peripheral blood cells in ECD and other histiocytosis. Welcome, Irena.
Good morning, everyone. I'm a pathologist and a PhD student, and I will present you today a part of my PhD work in collaboration with the French team about the distribution of MAP2K1 variants in PBMCs in Erdheim-Chester Disease and other histiocytosis. A few words about MAP2K1 mutations. They are actually common in histiocytosis. MAP2K1 gene encodes for a MAC protein, which sits just underneath the BRAF V600E on the MAP kinase pathway. And when mutated, mutually exclusive with BRAF V600E, it keeps the pathway switched on, leading to uncontrolled proliferation of histiocytes. MAP2K1 gene is the most frequently mutated in Rosai-Dorfman Disease, and the second most mutated in ECD and LCH after BRAF V600E. Maybe I can, yeah. It is known that there is a category of patients with clinical presentation of ECD and histology of Rosai-Dorfman disease, and they are commonly mutated for MAP2K1. We started to present interest into this gene recently because even if its overall phenotype overlaps with that of BRAF V600E, it is known today that patients harboring MAP2K1 mutations, they never develop neurodegeneration. And the reason for that today remains unknown. It's been shown that MAP2K1 mutations can occur in any age, any localization, in any type of histiocytosis. And while BRAF V600E is only one mutation, MAP2K1 can show over 60 variants in all types of histiocytosis. It's also been shown that the type of mutation differs with the type of disease. Single base substitutions are mostly associated with ECD and RDD patients, while deletion insertions are mostly associated to LCH. There have been also 3 functional classes of MAP2K1 described, first in 2018, based on the dependency of RAF upstream protein on MAP kinase pathway. Class 1 mutations are RAF dependent, and they have weak transforming activity. They respond to MAC inhibitors, compared to class 3, which are completely RAF independent with a strong transforming potential, currently unresponsive to MAC inhibitors, with worse prognosis, as we can see, than class 1 and class 2. And as it has been shown in this paper published by Eli Diamond this year, they are responsive to ERK inhibitors. It is important to notice that all 3 classes are found in histiocytosis. A few words about BRAF V600E. Its cellular distribution, as it is well characterized today in histiocytosis. We detect this mutation not only at the end stage of myeloid differentiation in histiocytes from tissue biopsies, but also in earlier compartments, in bone marrow progenitors, in myeloid compartments, monocytes, dendritic cells, and also in lymphoid compartment, T and B lymphocytes. In LCH patients, its highest mutation burden is found in monocytes. And it has been shown that after a long course treatment with MAC inhibitors, there is a switch of detection towards the lymphoid compartment, T lymphocytes. It's been shown also by Paul Min team work that the mutation burden in LCH patients correlates with the clinical risk. In Erdheim-Chester disease, although less data is published, BRAF V600E is detected in both compartments, myeloid and lymphoid. So BRAF V600E, it has much broader and dynamic distribution than initially thought, and that leads us to the question, what about the MAP2K1 mutations? What are their distribution in peripheral blood in patients with histiocytosis? So the objectives of my study were to investigate the distribution in PBMCs of MAP2K1 mutations in PBMCs in patients with histiocytosis and to correlate their detection with the treatment, disease activity. Also to improve the, in order to improve the understanding of disease ontogeny in histiocytosis and MAP2K1 mutation. We included 34 patients with MAP2K1 mutation confirmed on the biopsy samples. Most of them were adults from the French reference center, but also some children. The distribution was, we included 19 Erdheim-Chester disease, pure or mixed, 11 LCH, 3 Rosai-Dorfman disease, and 1 L group histiocytosis. We collected and separated PBMC from the plasma and then sorted the PBMCs into different populations. We studied the monocytes expressing CD14, myeloid dendritic cells, plasmacytoid dendritic cells, T lymphocytes. And even if I didn't show here, we also studied CD56 positive cells. Unfortunately, due to a technical problem, we were not able to analyze the B lymphocytes. We tested these populations by using digital PCR for MAP2K1 variants corresponding to each patient. I illustrated here on the structure of MAC protein, the mutations that we are capable to detect in our lab by using digital PCR. Our panel targets 20 MAP2K1 variants, covering most of our patients with histiocytosis. This technique is a high sensitivity detection method. And as I mentioned earlier, these are the single base substitution that we can detect, which are found mostly in ECD and RDD patients, while deletion insertion are here, mostly found in LCH. So these are our first results. We were able to detect MAP2K1 in PBMCs in most of the patients with Erdheim-Chester disease, with 68% being positive in at least one PBMC subset, while 32% were negative. In contrast, only one patient with LCH and 1L group was positive, while none of the patients with Rosai-Dorfman disease showed positivity. We also asked if this detection might correlate with risk organ involvement in ECD patients. And as you can see here, the positivity, the percentage of patients positive was similar, regardless of central nervous system or heart involvement. We then asked if this detection correlates with the treatment and disease activity from 23 blood samples of Erdheim-Chester disease. MAP2K1 variants were still detectable in periods of clinical remission. Most of the samples were from patients under remission. But it was also detectable during active disease, although not enough samples maybe were not analyzed. Similarly, MAP2K1 variants were detectable in periods of treatment with either MEK inhibitors, interferon, or no treatment periods. And in fact, most of the positive samples were from patients under MEK inhibitors. Regarding the distribution in the PBMC subsets, we were able to detect MAP2K1 in all populations, in both myeloid and lymphoid compartments, with the CD56 positive cells being the most frequently mutated, followed by the monocyte CD14 positive, with plasma thyroid dendritic cells being the least mutated. When looking at the mutation burden, variant allele frequencies were generally low, with a median value of 1.6% and the range between 0.1 and 41%. The highest VAF values we found in CD56 positive cells, followed by plasma thyroid dendritic cells, and the lowest values were found in T lymphocytes. Unfortunately, we did not have samples at diagnosis, so that limits our ability to assess any change before and after treatment in these patients. We also looked at the functional classes, at the classes of MAP2K1. Most of our patients fell into class 3 and class 2. And these mutations were detected in PBMCs. The detection was slightly lower in class 3 compared to class 2, 20 versus 50%, although this was not very significant. I will present an interesting case of a woman of 59 years old with a mixed Erdheim-Chester Rosai-Dorfman Disease with multiple organ involvement. Here there is a lymph node biopsy which showed typical RDD histology with large histiocytes exhibiting emperipoiesis, while the skin biopsy showed ECD histology with foamy histiocytes. And what makes this case interesting are the high variant allele frequencies that we observed. First in tissue biopsies, 22, 19% in NGS, which are unusually high for tissue biopsy of histiocytosis. Mostly the values are under 10%. And even higher values, VAF values in peripheral blood, with 41% of MAP2K1 in CD56 positive cells, followed by monocytes at 32% and plasma at 19%. It is important to say that the blood sample was collected while the patient was under complete remission, under trametinib for 16 months. And these are the highest values of MAP2K1 that we detected. And it is interesting, it raises questions such as why such high values persist in peripheral blood despite an apparently controlled clinical disease. And I will finish by showing our results in circulating cell-free DNA. We also tested the plasma of these cells, of these patients, and we obtained similar results to those in PBMCs. From 25 patients tested, 19 were positive, and the detection in cell-free DNA persisted despite MEK inhibitor treatment or remission periods, with no difference in VAF values. In circulating cell-free DNA, most of the VAF values were under 1%, and the values did not differ between active, partial complete response or no treatment versus MEK inhibitor treatment. In conclusion, MAP2K1 mutations in ECD are detected in both myeloid and lymphoid compartments, which suggest, as BRAVI600E, a common origin in an early precursor. These clones persist in PBMCs and circulatory cell-free DNA during remission or under targeted therapy. And we observed the highest VAF values in CD56 positive cells, which we will have to characterize further. They might be NK cells or maybe CD16 positive monocytes, a marker which we did not include in our panel. And we will also have to characterize the B lymphocytes also, PBMCs. I want to thank everyone who participated to this work, and thank you.